Okay, so we have discussed the passive ways of moving things across the membrane using diffusion or facilitating diffusion. Now we're going to talk about the active means, the active transport, the work being done to move things across a membrane. Now here, it's going to be an ion or molecule, no matter how small, like glucose, or big, like a protein or a chunk of a cell, has to get across the membrane. It cannot pass across the lipid bilayer on its own because of size, because of charge, because of whatever. So there's going to have to be some work done to get it across. Another thing that we find here is that unlike the passive mechanism, which was using high to low diffusion, what we're going to be seeing here is that with active, we're going to be taking things at low and we're going to be pushing them across the membrane to where it's high. Hydrogen, or electrons, is a great example of this. Our cells want to push this across membranes. They want to keep high concentration of hydrogen on one side of a membrane versus the other. It's going to drive a lot of our metabolic activities. However it's done, or for whatever reason it's being done, active transport will always require energy. Energy in the form of ATP in some mechanism, some way or form. We'll see that with active transport, the transport protein can utilize ATP directly. This is what's referred to as primary, or some other transport mechanism uses ATP to push, to drag some ion across the membrane from low to high concentration, and then the transporter we're paying attention to will then utilize that ion flowing back through it to pay the price to transport across some large molecule. By utilizing these transport mechanisms, our cells can set up imbalances. One side's going to have more of one type of ion than the other. We refer to this as an electrochemical gradient. It's higher on one side than the other, more charged positive on one side than the other means electrons are going to accumulate. One side's positive, the other side's negative. That's where the electrochemical comes in. By having positive ions on one side, negative ions on the other, well, it's basically almost sounds like a biological battery. Because think about it. You look at a battery you're putting inside a remote control or whatever. One side, one end's going to say positive, the other side's going to say negative. Well, that's what we have going on here. One side's mostly positive, one side's mostly negative. It sets up an imbalance that can then power other things. Now, when it comes to transporting, we're going to see that carrier proteins, these things that do the work, can either move one thing at a time across. Now, it may be multiples of, as you see to the left there, the uniporter moving A across. It could be multiples of A at one time. Or it could be something like a symporter, moving two different things at the same time, A and B. A may need energy to transport it across, whereas B's the toll that's being paid. Somewhere else there's a uniporter pumping B across the membrane. B's flowing back through the symporter, and by doing so it pays the price for A to come along with it. B could be a hydrogen. A could be something as large as a glucose molecule. Too big to diffuse across, So it's going to need an active transporter. And instead of utilizing ATP here, well, you're just going to utilize the electrochemical gradient that some other hydrogen transporter, some other hydrogen pump, electron pump, is pumping hydrogens across the membrane for. We also see antiporters. Going to transport, move A across the membrane, and in so doing, you're going to move B in the opposite direction. The sodium-potassium pump is a great example of this. You look here. You see the figure above my head of the sodium-potassium pump. It's an active transporter. How do I know? Well, because right here is ATP. Anytime you look at a figure and you're not sure if there's an active transporter or not, and it says ATP anywhere there, well, there you go. The active transporter, known as the sodium-potassium pump, this is the gold standard. We know everything there is to know about this. We've studied it. We know what genes are required to make and maintain the pump. We know under what conditions it's functioning. Basically, it's pumping sodium and it's pumping potassium. Sodium out, potassium in. But notice, sodium is already high outside, potassium's already high inside. So you're taking from low concentration and then you're pumping it across the membrane to where it's already high. This is the opposite of diffusion. You're pushing the ball up the hill. Diffusion's letting the ball roll down. It takes no energy. It's a natural state of matter. Here you're going against. So it's going to be work. It's going to take a pump. By pumping sodium out, the cell keeps as little sodium, free sodium, inside the cell as possible. And then the flip side, potassium. Keep as much potassium inside the cell as possible. This will have implications for other metabolic functions, but that's the role of the sodium-potassium pump. Now, as I talked about the secondary active transport, here they're talking about sodium moving back in. I may use an example of the hydrogen. Here, sodium moving in, sodium being a singular atom, takes a lot less energy to pump it out. Allow it to flow back in, and then this transporter you see right here. Transporter is just spinning. It's moving, opens and closes, or however you want to think about it. As the sodium that's been pumped out flows back through, it's allowing glucose to come in along with it. Glucose on its own is going to take a lot of energy, a lot of work, because it's a much bigger, it's a molecule, as opposed to sodium's individual atom. For the cost of pumping out one atom and having it flow back in, glucose, C6H12O6, 24, something that's 24 atoms inside, gets to flow back in at the cost of pumping out 2 sodium atoms that flow back in with it. It works for the cell.